All right, let's talk about bacterial cell components. And these are going to be cell components that are also present in the other types of microorganisms we're going to talk about, but we're primarily dealing with bacteria this semester. So I want to make sure that we talk about the components that are going to be really, really critical to you. Now, this is going to be one of our longer lectures. I hope a lot of it is review, but if it's not, take the time to pause each slide and take a look at what's going on. Understanding the cell components is really critical for understanding how we influence cells or potentially destroy cells as environmental engineers. So that's going to be really critical. So I know this one's going to be pretty long, so hold on tight. You can pause at any point and come back to this, right? So take your time, make sure you pause when you need to. All right, so in terms of bacterial cell features, we're going to talk about 6 different things. We're going to talk about the cell envelope, the cytoplasm, glycocalyx, appendages, endospores, and genetic information transfer. So we're going to see a lot of biology terminology, and it's always going to take me an extra second to pronounce, so bear with me here. So first is the cell envelope. So this is what surrounds the cell. You might have heard cell wall, cell membrane before, but when we talk about bacteria, it's the cell envelope. And basically bacteria are classified into major groups based on their cell envelope, and they can either be categorized as gram-positive or gram-negative. So gram staining is a procedure that has been developed in order to stain bacteria, and different bacteria show up differently. So basically the procedure is that we have a cell and we apply a specific type of dye to that cell, and we have a couple different steps. And based on what we see under the microscope, all these 4 different pictures down here on the bottom of this slide, we can figure out whether or not the envelope is gram-positive or gram-negative. If you want to know more about gram staining, there is a video in the class playlist that teaches you how to do a gram stain if that is something that you're interested in. But for the purposes of us in the class, what we need to know is that the differences in the cell envelope between the two are going to influence how we provide treatment or make changes as environmental engineers. So I want to show you 2 different cell envelopes. The one on the left is a gram-negative, and the one on the right is a gram-positive. And so you'll see that they have different structures. So gram-negative is going to have this bottom lipid layer, a cell membrane level layer that has membrane proteins in it. It's going to have this periplasmic space, and then a thin layer of what we call peptidoglycan, which is a type of protein, an outer membrane, and then these lipopolysaccharides. And these lipopolysaccharides in a gram-negative bacteria are the way that gram-negative bacteria interact with the environment. So if you think about maybe wanting to get rid of a gram-negative bacteria, interfering with its ability to interact with the environment is one way that we could potentially interfere with this cell. Gram-positive, you're going to see this membrane space, these lipids in the cell membrane, again with these membrane proteins, but you're going to see a much bigger peptidoglycan layer on top of that periplasmic space. And what you will see is there are teichoic acids and lipoteichoic acids on the outside, and these are slightly different cellular structures that a gram-positive bacteria would use to communicate with its environment. So there's a different way that we would have to inhibit this communication if the cell was gram-positive. So knowing whether a bacterial cell is gram-positive or gram-negative could help us potentially figure out what is the best way to deal with a cell, whatever that means in our specific scenarios. So as I mentioned on the last slide, your gram-negative envelopes are characterized by 2 membranes, inner and outer, with a thin layer of peptidoglycan. And they have lipopolysaccharides on the outside, and that's what mediates those cell interactions with the environment. Gram-positive envelopes have a single cell membrane interior to a thick layer of peptidoglycan, and they have different acids that stretch from the cell to mediate interactions with the environment. So the thing that I want you to take away from this is knowing the structure of your microorganism can be very important to understanding how we interact with that organism, either in a beneficial or a harmful way. Both gram-negative and positive cells have cytoplasmic membranes. So let's grab my laser pointer. They are impermeable to many nutrients the cell needs for growth and energy production, but they have these membrane-spanning proteins embedded in the membrane. So a membrane itself is made of a phospholipid. So it's called a phospholipid bilayer. You may have heard that before, where they have a hydrophobic head and a hydrophilic head and a hydrophobic tail. So basically what will happen is all of those heads will face outwards in the cell, and all of those tails will face inwards. And these membrane proteins allow the cell to selectively transport things in and out of the cell. So what this does is it's a protection mechanism for the cell. If not everything can get through, it can protect the cell from specific chemicals. But this is important to us as environmental engineers because if we want specific chemicals to go through a cell membrane to either lyse the cell or destroy the cell, then we need to know what that layer is. So both gram-positive and gram-negative cells are going to have that semi-permeable membrane. There is some other terminology that you might see as environmental engineers that I want to make sure that we hit before we talk about what its implications are. The first is antigenic, and this is something that causes an immune response. And then we also have the word endotoxic, which means that it's potentially toxic to humans and animals. So two different terms. Biology is a lot about terminology. So I recommend potentially keeping a little maybe vocab list for yourself might be a really good way to remember what these terms mean so that you don't have to keep coming back to this slides. And the relevance of those two terms to environmental engineering and the cell envelope in general is that generally, if we lyse cells, and to lyse means to break them apart, endotoxins from a cell can be released into the environment. And that's something we don't want. We don't want endotoxins in the environment. So if we are trying to get rid of a cell, we might not always want to lyse that cell based on what's inside of it, especially if it's an endotoxin. If we want to quote-unquote poison a cell, we're going to need a molecule that passes through those protein transports. Otherwise, that semi-permeable membrane is going to reject it. So we can lyse cells or we can poison cells. If we want cells to stop quote-unquote talking to their environment, we need to know if it's gram-positive or gram-negative so we can inhibit those correct molecules. And one thing that's kind of interesting from an environmental engineering standpoint is we actually have used an approach called biomimicry to create protein transport molecules. So those molecules that we see in the membranes, they have a very specific selective function, and we've actually tried to mimic that function in a lot of bioengineering applications, which can cross over into our field quite a bit. So these are some of the reasons that we care about the cell envelope when it comes to environmental engineering. Next component of the cell is called the cytoplasm, and this is that gel-like matrix that basically holds the rest of the interior of the cell together. It's composed of water, enzymes, nutrients, waste, and gases. It's basically that extra space that's holding all of those organelles and all of those components together inside. It's going to contain the ribosomes that are used for protein synthesis. In bacterial, it can have what are called auxiliary plasmids with RNA and DNA. So we'll talk about bacterial plasmids in a moment. And then the DNA in bacteria is found in the nucleoid in the form of the bacterial chromosome or in plasmids throughout the cytoplasm. So bacterial DNA is different from human DNA in that since the bacterial is most likely a singular cellular organism, it's in a specific nucleoid area or it's in those little plasmids. So let's talk about that really quick because genetic information is also important. So bacterial chromosomes average about 4 million base pairs and encode thousands of genes. And the bacterial chromosomes in the nucleoid contain the main genes for biological function. So the bacterial chromosome is actually pretty long by biological standards. It's 1.3 millimeters in length. It fits inside the bacteria because we have proteins, histone proteins, that will compact that chromosome. And so instead of having this loop structure, which you see on the left, having this orange circle, that's called a histone protein, it helps to fold the chromosome around it. So in general, the bacterial chromosome in the nucleoid, that particular piece of DNA is what's doing the main functions for the system. Bacterial plasmids are small circular DNA molecules that contain genes that's not mandatory for cell growth. So these are auxiliary genes. They're not something that's critical to cell function. But one thing that's really interesting about bacterial plasmids is that they can be obtained from other bacteria. So they can be retained if there's a selective pressure. Otherwise, the bacteria might lose them. So you're going to see a table of some different types of plasmids on the right side of the screen that were discovered and some different references if you want to take a look at bacterial plasmids in more detail. But the one thing that I will say about bacterial plasmids, that are important is that they can help transfer genetic information. And that's going to become important when we talk about antimicrobial resistance, especially in bacteria. So plasmids, as I mentioned, I always get ahead of myself, are how we confer antibiotic resistance to bacteria. So if a bacteria has a specific plasmid that is antibiotic resistance, it's actually very easy for that bacteria to transfer that gene to a different bacteria. And we can find antibiotic resistant genes in bacteria in wastewater. It's very easy to transfer to viruses and protozoa, and it's very hard to remove from the treatment, from the environment with typical treatment methods. It's very hard to remove DNA from wastewater and drinking water. It's easy to remove big particles, but it's hard to remove small particles. So that's why bacterial plasmids kind of have some relevance to us as environmental engineers. So to go back to the cytoplasm, the other component of the cytoplasm that's important is the ribosomes. The ribosomes are basically a protein factory. So basically what will happen is we will have messenger RNA, which is mRNA, which is bringing a message from the DNA to the ribosome. And then what that will do is it will link up with the ribosome and it will help the ribosomes make a protein based on what's in the code in the RNA. And remember that proteins are going to carry out your basic metabolic activities in cells. So it's really important to have these pieces of RNA going through the ribosomes to make the protein. So your ribosomes are your protein factory. Generally, ribosomes have what's called a large component, which is the 50S, which is this head up here on the top, and a small subunit, which is the 30S down here. And both are made up of ribosomal RNA proteins. As an interesting side note, 16S rRNA is a component that is found in bacteria and archaea that is actually used to classify organisms. So RNA has a lot of really important implications the deeper that you get into microbiology. For our purposes, it's good to know that RNA is involved in how we make proteins in cells, and that's probably good enough for us to stop with at this stage in the class, but we can talk about it in more detail as we need to. The next component I want to talk about is glycocalyx. And basically some bacteria have this interesting extracellular layer that's composed primarily of polysaccharides, but can also have proteins in DNA. So when you see polysaccharides, poly means many, SACCHA is a base pair from Latin that means sugar. So polysaccharides are carbohydrates. So basically what can happen in a cell is you basically get these like funky looking slime layers on the outside of bacteria. And the basis of what's going on in that particular layer is that it's protecting the cell from desiccation, so drying out, predation, being eaten by other cells, and chemical toxicity. So it can act as basically a repellent barrier. It can also help bacteria attach to surfaces, and that's very relevant to us because then these types of cells can produce biofilms, and biofilms are really relevant in environmental engineering. So biofilms, or sometimes we call them microbial mats, have been found to bind really well to metals, which makes them really good for binding and removing heavy metals from industrial waste. But it can also corrode drinking water pipes because our drinking water pipes are made out of metal. So microorganisms that have this glycocalyx can basically colonize an area of a piece of pipe or metal, and they exude what's called an extrapolymeric substance, an EPS, which has a lot of that glycocalyx in it. See, I told you, biology's hard because of the words. And then over time, it makes this biofilm. And that biofilm is going to interact with the metal that it's on. And it can potentially have some harmful side effects in our drinking water systems. So this is just another look at what's happening on the inside surface of pipes in our distribution systems for drinking water. And this is becoming one of the main kind of evolving concerns in the drinking water industry right now, because we have to worry about the quality of our water, not only after we've treated it, but the quality of our water in our pipes. So that's some relevance of glycocalyx to environmental engineering. Fourth component we're going to talk about, moving right along, I'm trying to keep it short, are appendages. And these are basically accessory structures that extend from the cells. Most of the time they are involved in what we call motility, which is the ability to move around. So we can have what are called flagella, which are these long, like whip-like structures that are normally 1 to 2 per cell that help kind of spiral a cell through an area. So many times they have helical motion. We can have fimbriae, which are basically kind of these spinny little threads that are coming off this picture here. And they're really about attachment. They help a lot with biofilm formation, which again, biofilm formation is good from a bacterial standpoint, not so great in a lot of our built environments. And then we can also have what are called pili. And so that's this structure that's connecting these two cells here. And basically it's for a mating process to be able to transfer DNA between bacteria. And appendages are really about motility, right? Moving around. So you can have what's called positive motility, which is moving towards nutrients, or you can have negative motility to move away from harmful chemicals. So what's on the left here is a happy bacteria moving towards its nutrients. That's positive motility. Or you can have negative motility, an organism kind of fleeing away from these harmful chemicals. And your appendages are also great for attachment to surfaces. So sometimes we look at these structures when we're worried about biofilm formation. Fimbriae, as we mentioned, aid in the attachment of cells to surfaces. Pili are essentially the same as fimbriae, but only found in gram-negative bacteria, and they're involved in that mating process through conjugation. We'll talk about conjugation later in these slides. All right, moving right along. 2 more to go. I promise. Stay with me. Hit pause if you need to. Number 5 is endospores. So some gram-positive bacteria in particular produce multi-layered structures that are capable of withstanding adverse conditions. So these are endospores that are shown in this picture, and they're kind of hard to see. But basically, it's a survival mechanism for the cell. So if the cell is exposed to radiation, to UV light, heat, desiccation, low nutrients, any of those scenarios could cause a bacteria to produce endospores. And it's basically a structure not really well studied, honestly. We're still learning more about it. But it allows bacteria to survive for long periods of time in a dormant state until they encounter the right conditions for growth. So it's a survival mechanism. It's very, very important when we talk about environmental engineering for the survival of pathogens and disinfection procedures. A lot of pathogens, which again, remember a pathogen is something that's infectious to another organism. Remember that those we want to be very careful about when we try to disinfect, especially in water or wastewater treatment. And if pathogens are hiding inside an endospore, they're protected from whatever chemicals or exposure we're using for disinfection. So if we have bacteria hiding in those endospores, our disinfection may not be all that super effective. And so this is becoming an area we're studying more and more in order to understand how we need to do disinfection a little bit better for these types of bacteria. The last thing that we're going to talk about is genetic information transfer. And we're going to hit this at a very, very high level because this itself is a whole field of study. But basically, bacteria are amazing at adapting very quickly to changing environmental conditions. They have rapid growth and a very flexible bacterial chromosome, right? So we as humans have a massive genome, and as a result, we're not as flexible, believe it or not, to changes in our environment, not in the way bacteria are. So that rapid growth and that flexible bacterial chromosome are great because bacteria, they adapt like you wouldn't believe. So climate change, they're not worried, right? Microbial disinfectants, antibiotics, in many ways, they're not worried because they're very flexible, right? That's why we're getting antibiotic resistant microorganisms, right? So most of the time when a bacteria reproduces, it's through binary fission. And then there's this other term that we care about called homologous recombination. And this is a process by which bacteria incorporate new DNA into their genome. And it can occur by what we call lateral or horizontal gene transfer mechanisms. So we're going to talk through some of those mechanisms. There's 3 methods of gene transfer. The first is conjugation, which is direct cell-to-cell transfer of conjugated plasmid DNA through a protein pilus. So those pili structures that we saw in appendages, that is conjugation. Basically, there's a connection between the 2 cells, they transfer that DNA, and they're good to go. Then we also have transduction, and this occurs due to the accidental packaging of cellular genetic material during bacteriophage replication inside a host cell. That's a mouthful, so we'll talk through what that means. And then there's also transformation, which occurs when a bacterial cell obtains DNA from some surrounding medium. So let's talk through each of those at a high level. Conjugation is that pilus extending from a donor to a recipient cell, and it's generally plasmids that are being transferred for a variety of non-essential genes. So this is not necessarily the entire bacterial chromosome, it's those small circular plasmids. So this is how we get that antibiotic resistance in cells. So I'm not going to say much more than that other than it's direct cell-to-cell contact. Transduction is the funny one that's a little bit hard to understand sometimes, but basically what will happen is a bacteriophage will attack a cell. So a bacteriophage is a virus that infects bacteria. And so that virus itself is going to have that pink DNA and the bacteria is going to have the green DNA. When that bacteria attacks the cell, it will break up that genetic information. It's going to hijack the cell and basically use the cell's ability to replicate DNA to grow. So what will end up happening is as that bacteriophage multiplies and kills the cell, it's mostly going to be the original bacteriophage with these pink chromosomes, but it is possible that it's also going to take in these green chromosomes as well. So bacteriophages are going to end up killing the cell, right? They've infected the cell, so they're going to lyse the cell and they're going to get out into the out into the medium. So the new bacteriophage with the bacterial DNA can go and infect another bacteria, insert its DNA, and this bacteria can actually incorporate, though it's kind of hard to see, incorporate some of that new bacterial DNA. So what's happened is a virus has done some infecting, but it's also gotten some new DNA as a result of infecting, and then it transfers that new DNA to another bacteria cell. So it's spreading the genome even though the original bacteria cell is dead. Last is transformation. So transformation, we're going to have cells potentially dying, and when cells die, many times they lyse. And remember, lyse means break apart. So the cellular components that were inside the cell end up in the environment. And it's quite possible that another cell, which we call the recipient cell, can take up those different pieces of DNA. So that is transformation. There are DNA pieces, there's RNA pieces floating around in the environment, and for any reason, this particular cell will take that up. So that's important. DNA uptake by this method is random, and I want to point that out. It's not that cells are necessarily looking for DNA, so this is a random process. Last thing to consider is a question for you at the very end of this lecture. Which method of information transfer do you think is the most relevant to environmental engineers? And we will talk through this answer in class, but give it some thought. Write down your answer and let me know what you think when we get back into class. I know that was a long lecture, so thank you for staying with me to the end. I hope a lot of that was review. I hope it was interesting and it relates to what you're thinking about doing as an environmental engineer. And if you have questions, remember that you can always go back through the slides, pause, and take some extra notes. I will see you in class.